control, and the body's pH range is between 7.35 and 7.45, all right? With, of course, the average being at 7.4. That's the ideal range for the function of proteins in our body. So we have some molecules, some chemical buffers that the body uses, and then we have a couple of systems. Respiratory system we've already talked about, and the urinary system we're going to go over tonight with hydrogen and bicarb function. So inside, and this is interesting because one of the major buffers within a cell is the phosphate buffer system, but we're going to see it as a major buffer system in our filtrate, which is actually extracellular fluid, okay? Most of the major buffer system in extracellular fluid in blood is our carbonic acid system that we've been talking about for a couple of weeks. And proteins can be in both, all right? So hemoglobin is a major intracellular protein for our red blood cells. And we have other proteins in the blood and different amino acids that will function as a buffer. So let's look at some of these chemical buffer systems. Then we'll review the respiratory, and then we'll look at more specifically how the proximal convoluted tubule adjusts the movement of hydrogen or bicarb. So we're going to get 6 questions tonight. We started with 3 that last night didn't have. And we haven't discussed this yet, but it leads us into our discussion for the next component. So we're looking at an amino acid. The R here stands for the rest of the amino acids, and remember, proteins are like beads strung together. And so the R represents the rest of the amino acids strung together in this bead. So let me write that up on the board. So we have nitrogen, okay? And nitrogen has a couple of hydrogens plus a third bond that it shares with carbon. And the rest of the amino acids will start up here. And then a hydroxyl group. So there's 2 places where hydrogen can be, all right? We can have an extra hydrogen here. So has this donated or accepted hydrogens?
Accepted.
Accepted them. So when do we accept hydrogens?
Acidic.
An acidic condition or an alkaline condition? So we have too many hydrogens in the floating around, and when we don't have enough. Acidic, when we have too many. This is not done by enzymes, it's not done by hormones, it's purely the number of free hydrogen ions floating around. So if we have a lot of hydrogen ions, so many that we're in acidic condition, it's going to force one on nitrogen, so this will have an extra charge, and our hydroxyl group will have its hydrogen. So what would this look like in a basic situation where we need to donate hydrogens? We certainly don't have this one. And even this one will come off, all right? So that's what an amino acid or protein would look like in a basic condition where it had to donate hydrogen ions to buffer a low pH. Or, to buffer a low number of hydrogens, a high pH. All right, so you would see this in a basic or alkaline condition. Is that starting to make sense? I don't want it to be something you just memorize. I want you to recognize how adding or subtracting hydrogens would buffer the number around the situation, okay? So you have this slide on your lecture PowerPoint. You can go back and review for whether it shifts from releasing to accepting, depending on the pH of the surrounding solution. All right, we're not going to do this one on your PowerPoints because we've done 6 already, right? So let's just look at these to answer them. You don't need to put them on your Scantrons. So there's carbonic acid, which we've already described as the relationship between water and carbon dioxide. So if it's a basic condition, would we see it in this form or in this form? The bottom one, where it's donated its hydrogen, okay? And if it's a acidic condition, the hydrogen is gonna bind to this, take that hydrogen out of solution, okay? We're gonna see that with the urinary system as well. And here we are. Now, before we talk about the urinary system, let's review what happens with the respiratory system. So let's say that you're in ketoacidosis, okay? Which means too many hydrogen ions from breaking down fatty acids for energy. Fatty acids are acidic. They're going to release hydrogen ions. And so in ketoacidosis, we have a low pH. Another situation is diarrhea. Now we haven't done the digestive system yet, but I just mentioned that the pancreas releases bicarbonate ions to buffer the 1 to 2 pH level of the gastric secretions. So if you have a lot of diarrhea, then you're getting rid of those bicarbonate ions and not absorbing them. So now you've absorbed hydrogen from the stomach without the buffering bicarb, okay? And the opposite effect, if you have a lot of vomiting or emesis, you're losing the hydrogen ions that would have buffered the bicarb released by the pancreas. So that would be alkalosis. But in diarrhea, there's a loss of pancreatic bicarb, too much of the gastric hydrogen ions. Okay? So low pH. What would happen to your respiratory rate? When you breathe, we're not worried about the oxygen right now, okay? Carbon dioxide. So are you gonna hold on to your carbon dioxide, or are you gonna breathe off more carbon dioxide is the question. Because what happens with carbon dioxide? Carbon dioxide has no hydrogens. So why does it change our pH? Because some of it, a lot of it, most of it binds with water to form carbonic acid. This is where we get or remove the hydrogen ions with respiration. Because we remove or keep with deep respiration and rapid respiration, we get rid of CO2, which means it cannot bind with water. Therefore, we do not make carbonic acid. Therefore, this does not dissociate into hydrogen. So even though we're not actually losing or gaining hydrogen, we affect the direction of this formula, okay? So for this one, do we need to get rid of CO2 or do we want to keep CO2? If we keep CO2, we're going to form carbonic acid and we're going to add hydrogen ions as that dissociates, okay? So somebody who has pneumonia, and we've increased the diffusion distance, and CO2 builds up in their blood, their pH is more likely to go down. So in this condition, we want to breathe more rapidly and more deeply. Does that make sense? Because the less CO2 we have, the less carbonic acid we make, the less hydrogen is going to dissociate, okay? So in metabolic acidosis, respiratory system is going to increase respiratory rate and depth to get rid of CO2, so we don't make carbonic acid, okay? Now, if the problem is respiratory, we can't change it by changing our respiratory. We have to do our metabolic changes, our urinary system, okay? But if it's a metabolic problem, you can see some changes with respiratory. All right, so let's say now we're in metabolic alkalosis. Our pH is too high, vomiting or emesis, or an overdose of Tums, all right, which is an alkaline chemical to neutralize the pH of the gastric juices. So in that case, our pH is too high. So now we want to keep some carbon dioxide so that we can contribute these hydrogen ions to to bring the pH down, all right? So in this case, you are going to decrease the number of respirations and the depth so that you retain carbon dioxide. However, you're not going to fix any pH imbalance by not breathing at all, okay? So there's a limit to how slow you can lower your respiratory rate and still get adequate oxygen. But this is how the respiratory system, as a system, keeping or getting rid of CO2 can be used as a buffer. Even though we're not actually getting rid of hydrogen or keeping hydrogen, we're affecting the hydrogen associated with the carbon dioxide. Does that make sense? Okay? So if you're not breathing, you're keeping the CO2, you're making more carbonic acid, more hydrogen. If you are breathing more deeply, you're getting rid of the CO2, which in effect is getting rid of the hydrogen you might make from that CO2. All right, well, let's look at what we do with the urinary system, because if we have a respiratory cause for acidosis or alkalosis, we're not going to be able to use the respiratory system to fix it. All right? So we're going to go back to the proximal convoluted tubule. And remember I said we were going to talk about secretion? Here's where we're going to do a little bit more of that secretion. And secretion is in which direction? Blood to filtrate or filtrate to blood? Blood to filtrate. Okay, so I'm going to draw that simple cuboidal epithelial cell again. The very long microvilli. Filtrate is going to be here. And this is in proximal convoluted tubule. Okay? So our plasma has too many hydrogen ions. Our pH is low. Hydrogen ion concentration is high. So we want to end up by moving the hydrogen into our filtrate, all right? We want to go from peritubular blood to filtrate, and we call that secretion. Should be simple enough. So we take our hydrogen ion and we send it towards the filtrate. It won't cross the basal membrane of our simple cuboidal cell. Where did we see that before? Where hydrogen ions wouldn't cross the epithelium?
The central chemoreceptors.
Central chemoreceptors, the cells lining the fourth ventricle of our cerebrospinal fluid. So what did we do instead? What did we send across the epithelial tissue since the hydrogen ions wouldn't go?
Carbon dioxide.
We sent gas. We sent the carbon dioxide gas, okay? So we take hydrogen ions plus bicarbonate ions, and that makes our carbonic acid, which we can get our gas from by dissociating it to carbon dioxide. That will now diffuse across into our simple cuboidal cell. Within that cell, carbon dioxide reverses the process, interacts with water to form carbonic acid, and it does so under an increased rate with Ca. What's Ca? We found this in red blood cells. We found it in the cerebrospinal fluid. It's not in the plasma, but now we find it in our simple cuboidal epithelial cells. Do you remember what Ca stands for? Carbonic anhydrase. So this is going to increase the rate of the reaction of the water and the carbon dioxide back to our carbonic acid. That will dissociate to form our hydrogen ion. And we're going to return the carbon dioxide back to the plasma, which we need to buffer because we have too many hydrogen ions. We want to send that bicarb back. All right? The hydrogen then gets pumped out with sodium. So this takes ATP. The sodium doesn't go out on its own, right? So the hydrogen gets pumped out. Sodium gets pumped out. And now we've added, secreted hydrogen ions into the filtrate. So some of them got filtered, some of them didn't. And now we've secreted some of the ones that didn't and added them back to our filtrate. So now our plasma has fewer hydrogen ions than it did before filtration, all right? So we have hydrogen ions in our filtrate. What's going to happen to the pH of our urine? It's going to drop, all right? Burning urine at a pH of 1 or 2, hydrochloric acid, it's not going to feel very good, even if we have transitional epithelium. So we have 3 buffer systems that work within the filtrate itself. Remember I said phosphate was typically an intracellular, but we have a phosphate system here, HPO4, except an extra hydrogen. Our ammonium ion is formed, and carbonic acid again. And that kind of forms a sink where we can keep adding hydrogen to the urine. And it's usually around a pH of 5 when it's really acidic, okay? So between 5 and 6.5 or so is typically our urine pH range. So, in addition to secreting hydrogen ions, we can move bicarb that was filtered back and add more bicarb to our plasma. So not only can we get rid of hydrogen ions in the blood, we can add bicarb back that was filtered to provide more buffering bicarbonate ions in the blood. Okay? So that's going to be reabsorption. We're going to move from the filtrate back to the blood, and we do bicarbonate reabsorption. Okay? Oh, we got a problem. Bicarbonate ions won't cross the microvilli. Well, we do the same thing we did in the blood. We take a bicarbonate ion plus a hydrogen ion, and we turn that into carbonic acid, into water, and carbon dioxide, and we send that back into the simple cuboidal cell. Use the carbonic anhydrase again. Return the hydrogen from the water that we used and add more bicarb back to the plasma. So now we've doubled the effectiveness of the urinary system as a buffer, because we got rid of hydrogen ions, plus we returned a bicarb that was already in the filtrate back to the blood where we can use it to buffer. Okay? So that's using our chemicals, but within the urinary system. So this works if the body is in acidosis, and we're trying to get rid of hydrogen. If the body is in alkalosis, will this work? No. So if the body's in alkalosis and the pH is too high, this just stops. All right? We just don't even use this, because this is only, proximal convoluted tubule is only for acidosis. However, remember the collecting duct? I'm not even sure if I can find the slides quickly enough again. But in the collecting duct, we have the intercalated cells that had the receptors for ADH. Okay? In addition, may I erase the board here? Anybody do pictures of that or anything? Okay. So in the collecting duct, this is not hormone driven, this is all numbers of ions. We have alpha and beta cells. And the alpha cells of the collecting duct work just like the proximal convoluted tubule. Hydrogen ion secretion and bicarb reabsorption. And I'm all for memory aids. So think of alpha A for acid. Okay? So under acidic conditions, the alpha cells are active and secrete hydrogen ions and absorb bicarb. Under under basic conditions, the beta cells in the collecting duct, we don't have anything comparable in the proximal convoluted tubule, are going to secrete bicarb and reabsorb hydrogen ions. So they do the opposite of what the alpha cells and the proximal convoluted tubule cells do. So let's look at a situation and see what the respiratory system and the urinary system are going to do together. Okay? And then we'll be done with the urinary system's component of acid-base balance. We'll take a break and come back and look at how to do the homework on figuring out pH and bicarb levels in the whole Henderson-Hasselbalch equation. Okay? Which is not as difficult as it sounds. So without thinking about whether the cause is respiratory or metabolic, just the pH condition. If the pH is less than 7.35, the respiratory system will do what? Increase or decrease respirations? Increase. So respiratory rate will increase. And respiratory depth will increase. Loss of CO2, okay? Meaning less carbonic acid. Urinary system. In an acidosis, are the proximal convoluted tubules going to be secreting hydrogen ions, or are they going to be inactive? They're going to be secreting, getting rid of hydrogen ions. So our proximal convoluted tubule cells and our alpha collecting duct cells are going to be secreting hydrogen ions and reabsorbing bicarbonate ions. Okay? And the beta cells will be inactive. Okay? Now don't memorize that. That's something you should be able to figure out if you, well, you can memorize what the cells do, okay? But you should be able to figure out whether you're trying to keep hydrogen or get rid of hydrogen based on the pH. Okay? So now what is the respiratory system going to do? if you are in alkalosis. Not enough hydrogen ions. It would be the opposite. We're going to try to retain carbon dioxide in order to make more carbonic acid. So respiratory rate is going to decrease. And respiratory depth is going to decrease. So fewer and more shallow breaths, holding on to the CO2. Okay? So this would be retaining CO2. All right, with the urinary system, do we want to get rid of hydrogen ions?
No.
No. All right. So the proximal convoluted tubule cells and the alpha cells of the collecting duct are going to be inactive. They're not going to be secreting hydrogen. We certainly don't want any more bicarb back because we already have too high a pH. But now the beta cells will be active. Okay? And they'll be getting rid of bicarb and retaining hydrogen ions. So this will be secreting bicarb. And reabsorbing hydrogen ions. And that will lower our pH. Now, unfortunately, life isn't this simple. Because you can have respiratory acidosis and metabolic alkalosis, and then it gets all intercalated. So if you go to respiratory therapy, you learn about all these different overlaying complications. You can have somebody with emphysema and ketoacidosis, okay? All right, so we'll stop there as far as looking at body systems. Take a break and let me know when you're ready for the quiz, the histology quiz. And then when we're done with that, we'll look at how we figure out how the ratio of hydrogen and bicarbs helps us determine pH. I will be uploading the urinary lab. I'm probably going to combine that with the acid base into one lab, but you do have an extra credit lab at the end of your lab packet that's not online, and we're going to look at how to fill that out. Okay? It's essentially just kind of putting numbers into a formula, but I want to go through that with you. So let me know when you're ready to take your quiz. All right? It covers the microscope image of the glomeruli, the parietal layer, visceral layer, EMs of podocytes and filtration slits and so on, as well as recognizing the urinary pole, the vascular pole. If you see the proximal convoluted tubule leaving, that's the urinary pole. If you see the afferent arterial or any blood vessel, that would be the vascular pole. Recognize the macula densa by the close grouping of the nuclei, the granular cells, because you can actually see dots. What did we call the cells in between the podocytes and the capillaries? Intraglomerular mesangial. Okay. It's hard to recognize them, but if I say name the cells that are between that, and then be able to tell the difference between proximal and distal convoluted tubules, between blood vessels like the vasa recta versus non-blood vessels like the nephron loop thin segment. So if it has cells in it, it's a blood vessel. What would it be if it was in the cortex and it had a simple squamous wall and blood cells? Would it be vasa recta? No, it would be peritubular capillaries. How would you know it was in the cortex? You would probably see the capsules, and you would see proximal convoluted tubules, okay? If it's in the medulla, you're not going to see those capsules, and your collecting duct cells are going to be very tall, very columnar, okay? That's about it. So let me